Hello, and welcome to my poster about the high-performance annotation of biological sequence profiles. I am Martin, and I will be walking you through the poster content reorganized into a slide display for your convenience. Let's start with the background section. Functional annotation of biological sequences relies on identification of sequence homologs. Hidden Markov model and position-specific scoring matrices are commonly used to model prior knowledge of sequence families. Position-specific scoring matrices can only model gapless conserved regions, whereas hidden Markov models are able to model entire domains, including insertions and gaps. To support annotation with these two models, we developed high-performance packages to improve the throughput of sequence annotation using different optimization techniques. Let's start with the first package, named PyHammer. PyHammer is our Python package providing bindings to Hammer, a popular tool for sequence annotation using hidden Markov models. The package features a rework dispatcher for running many-to-many comparisons with Hammer on several CPUs using multi-threading. Basically, as shown below, the PyHammer dispatcher will attempt to maximize occupancy of the worker threads by reducing synchronization requirements, where Hammer would only start launching a new query when the previous query was finished. This greatly improved the performance of PyHammer on several threads, as shown in this slide, where basically PyHammer is able to diminish the runtime greatly with only 8 threads and reaches a speed up of more than 30 times faster when using 64 threads instead of 1 thread. Modeling the speed up according to Amdahl's law actually suggests that PyHammer runs up to 96% of the code in parallel sections, where Hammer can only run 35% of the code. This is actually very interesting when running genome size annotations, when the target database is composed of roughly a couple of thousands of proteins, when several threads are available, PyHammer is going to greatly improve the performance speed. We tried running a large-scale annotation of more than 2,600 bacterial genomes using pfam with 6 threads, which is a typical number of threads for a modern consumer machine that has 4 to 8 CPU cores. And on this particular task, PyHammer was able to make the annotation 3 times faster on average for each genome. And in total, it actually took only 27 hours, where Hammer needed 97 hours to complete. The other package we developed is actually a package called Leitmotif, for running position-specific scoring matrices scoring. This actually is using a platform-accelerated implementation that we implemented from scratch using Rust, and then provide binding for Python. This allows the parallel scanning of independent position by reorganizing a sequence. So basically, instead of keeping a sequence to be scored in a linear fashion, we reorganize this sequence into a matrix format, where each column of the matrix follows itself sequentially, and this allows each row of the matrix to be linearly independent. We can then use fast CPU extensions such as AVX2 or Neon to quickly scan the positions of every row of the matrix in parallel, allowing up to 32 columns to be processed at the same time in AVX2 or 16 columns in Neon. And this greatly improves performance and allows to compute the scores for the entire sequence given a PSSM in a much shorter time. We actually compared our implementation against other existing packages for scanning with PSSMs, where we found out that our package massively outperformed the other packages, as shown here, where Leitmotif can annotate up to hundreds of megabase pairs per second on a single CPU core. And it only takes a couple of seconds to annotate bacterial genomes with more than 400 PSSMs from the product database, where BioPython, for instance, will take more than 80 seconds to complete. Overall, these two packages should be really helpful for the large-scale annotation of sequence and they are actually compatible with existing standards, either using PSSMs from Transfac or using HMMs for HANA. I thank you for your attention. I hope you enjoyed the presentation. Please feel free to contact me at any time, or to actually have a look through the GitHub repository of both packages, which you can find on the poster itself. Thank you.